So the topic for this video is how do we know how many bacteria there are in a culture? Obviously it's very difficult and conceptually meaningless to talk about how many bacteria there are on the surface of a plate and count individual colonies, but when you have a liquid culture, obviously there are going to be on average a certain number of bacteria per milliliter. And knowing that is often important if we want to know how badly contaminated something is, if we are investigating what's the best growth medium for a particular type of bacteria, if we want to find the best temperature, whatever, for growing our bacteria. We're going to want to try various things and then find out how many bacteria per milliliter in our culture. Now, there are classically 4 ways of doing this. One is simply you get a very fine filter your liquid, bacteria are left in the filter paper, you dry the filter paper, you weigh it. That gives you an idea of how many bacteria. That obviously only works when there's a very large proportion of bacteria in the culture. Now, actually, one of the commonest ways in labs today when we're testing things is to use a machine called a spectrophotometer, which quite simply just shines the light through and looks at how much light has been absorbed, what percentage. Since bacterial cultures are all murky and bleh, then obviously the more bacteria there are, the less the light gets through. And in fact, when you go into commercial labs testing antibiotics, disinfectants, best growth media, that's what they do. Now, we do have some spectrophotometers, but they're not really for microbiological use, obviously. I don't want to put bacteria in case I contaminate the spectrophotometers, which are not designated as biohazard, so we won't actually demonstrate that. But basically, you just take your culture with your bacteria in, put it in a called cuvette, a little clear container like a mini test tube, put it in the spectrophotometer and a number comes up which tells you how much light is currently getting through the culture. The less light that's getting through, the more bacteria there are. Simples. Now, the third way is to test what we call a viable cell count. And I've demonstrated that, I hope, on another video. So I won't come back to that. That is in the video on finding the best growth media. It's the one where we dilute the culture 1,000,000 times, we've just got few bacteria, spread it on a plate, leave it, come back, and one bacterium has become a big blob, which isn't a science word, so we'll call it a colony, and you count the blobs, colonies. You know, 1, 2, 3, 4, 5 blobs. Well, that means there were 5 bacteria. I diluted it 1,000,000 times, so before I diluted it, there were 5 million bacteria. Great. And finally, and most obviously, you can just count them. That's what microscopes are for, after all. Put your bacteria on a slide, peer down the microscope and count them. Not actually as easy as it sounds. For one thing, looking down a microscope and going, oh, there's lots down here. 1, 2, 3, 4, I counted that one. Is more difficult than it sounds. And of course also you have to add some sort of dye that will show up the bacteria. It works better for things like human blood cells or large microorganisms like yeast that you can really see. But anyway, ways have been found to get around the, have I counted that one problem? And that is this special slide called a haemocytometer. As you can see, it's way thicker than a normal microscope slide, and it has sort of H shape on it. I know you can't really see that from there, and while I'm on my own I can't zoom in because this camera doesn't come with a remote, so I'll just draw you a little sketch. If you look at the slide, top on, you will see there is, always draw diagrams with a ruler, which I used to say. Anyway, you will see there is an area cut out like that. So this H shape is like down, it's incised. No, you can't see from there. Oh well. And in fact, although you can't see it with the naked eye, in here there is a tiny, tiny little grid drawn on, where the whole grid is like 1 millimetre, and you just can't see it. It is there, but you really can't see it. Now, the idea is, if you actually looked at this slide sideways on, what you would see is you have these deep cut-away bits, like that. But this bit and this bit is actually this little bit lower. So when you put the cover slip on here, there's like a space in there to put liquid in, where I've coloured in black. And that space is of a known volume. It's normally 0. I don't know what it is. It's written on the box. Anyway, so what do I need to show you? I just need to show you how to load a spectrophotometer. As you can see, I'm not wearing goggles. I haven't lit my Bunsen because I'm not using actual microorganisms. I'm just going to use water. There's a special way. It's not going to be sterile, so I'll just put that on the table. There's a special way of loading a haemocytometer, which I'm supposed to teach you. As I say, normally we would test different cultures of yeast. Unlike a normal microscope, you have to use a special cover slip. It's still glass, it's still thin, but it's maybe not quite as thin as a normal cover slip. It's also very expensive, and if anyone breaks one, the lab techs will not like me much. So, unlike a normal microscope slide, you put the cover slip on before you put the specimen on. And we want just a tiny, tiny, tiny bit of moisture to make it stick. Running it under the tap, that's too wet. So we have a really high-tech method of getting a tiny bit of water on. Like that. Okay? And then what you do is you put the cover slip down, sort of here, and you push that way. Push is the operative word. You have to press down really hard, which is why the slide is much thicker than a normal slide. So, okay, again, I know it's difficult to see a demo from there, but if you were in the classroom, you'd be sitting at the desk going, what is she doing? Anyway, so it's like this. And then I press right down, slide my cover slip into place. I need to check I've got a seal. I can tell because if I pick this up and turn it upside down, it falls off. That tells me I haven't got a seal. Let's try that again. Talk among yourselves. Not very sterile, blowing on it, but then whatever's going in the haemocytometer doesn't need to remain sterile anyway. That's better. Right. Also, you can kind of see what they call Newton's rings. I never quite know what causes them. Have little rainbows to tell you it's stuck. If you were in the lab, you could see pretty rainbows. Right. Anyway, that's stuck. Now I need to fill this space that I've coloured black with my sample, which would normally be my bacteria. In this case, I'm just going to use some water. Again, I'm not troubling to be sterile. And so how on earth do you get the water on when you've already got the cover slip on? It's actually easier than it sounds. Literally, all you have to do is put your syringe. I've now got my cover slip on. Hang on. I've now got my cover slip on. So my cover slip is now on. It's here. And all I have to do, it doesn't sound as if it would work, but it does, is put my pipette there, touch it to the edge of the cover slip, and let a tiny drop of water out. Like so. And to everyone's amazement, it sucks itself under. I would generally advise only doing one side of a haemocytometer. If you do two, the cover slip sometimes floats off and then it's come loose. But that haemocytometer is now actually full. And when I look down the microscope, what I will see is a tiny grid, typically 4 by 4, each section of the grid, 4 squares, that is, like so. Yeah, and that makes it much easier if my bacteria are scattered everywhere, or whatever other type of cell I'm looking at. Then it's much, much easier because I can count one square at a time. I can say how many bacteria in this square? 2. In this square, 1. In this square, 3. And so on. And I can much more easily record them without getting confused as to which ones I've already counted. Okay, I know, as I say, there's not a lot you can see from that distance. It's something that's better to learn by doing, but this is probably the best. I suppose if I take the focus lock off and come around there and point the camera at it, it's just possible the camera will pick up the haemocytometer. I'm not holding my breath. Okay, autofocus. Hey you, autofocus? No, you don't want to. Oh well. I don't think this is going to work. I don't even know if that's in shot because, as I say, my screen is over there. But there's my haemocytometer. Okay. Haemocytometer. Bye.